Sorry, I'm not feeling well this morning. However, I do want to make sure that we communicate with each other concerning the expectations for the rest of this quarter, which is going to end on April 5th. When you get back, there's going to be some assignments due. They're all in Schoology. Uh, Today's lesson, I was planning on going over alternative methods of inheritance, and I did do this with the first period class, so I would like you to watch and take notes on that lesson, and then continue to watch and do and take notes on the rest of the activities that are due on April 5th. If you have any missing work, please turn it in to into Schoology before April 5th so that I can give you the best possible grade you can get. I hope you have a relaxing spring break. If there's any issues, please make sure that you message me and I'll try to get back to you as soon as I can. I will be busy this spring break, however, so you might have to be patient and repeat your requests for your questions if necessary. I hope the video helps you. I hope everything goes well. You will have an exam on the second session of the week when we get back from spring break. It will be on all the materials we covered thus far. You have all the support you need on Schoology to prepare for the exam. Good luck, and if you need anything again, just message me through Schoology. Have a good day.
So again, I go through and I explain what gene linkage is in the videos and what independent assortment is again. I think the kids and I did a pretty good job at coming up with the answers and explaining how to do it. So I'm not going to talk about that today. What I'm going to talk about is the last part of this part here, this discussion part. Do you see this discussion part at the end of your Dragon Genetics Part 1? Can someone read this for me while I drink my coffee? Because it's early. Someone read it for me. Cheyenne, would you mind? Is it too small? All right. You can go on your Schoology and open up your document if you want. Do you see it now? It's in Dragon Genetics Part 1, right? Yeah, after discussion. Oh, you're looking it up. Somebody else read it, or no one else can read it either?
I'm reading the whole thing.
Mm-hmm. Where it says after discussion. Make sure your mic is on, though. Let me turn my mic off so that you can speak.
The principles of inheritance in these dragons also apply to inheritance in humans, other animals, and plants. However, inheritance in humans, other animals, and plants is much more complex than inheritance in these dragons. First, list the two principles of inheritance illustrated by the dragon genetics activity. What are some additional complexities of inheritance in humans, other animals, and plants? Discuss the following questions. Genetic linkage is very strong for genes which are located close to each other on the same chromosome. What happens in the case of two genes which are far apart on the same chromosome? Are all alleles either completely dominant or completely recessive? Do any genes have more than two alleles? Does each gene influence only one phenotypic trait? Is each phenotypic trait influenced by only one gene? For the genes that are on the X chromosome in humans and other mammals, what are the differences in inheritance for males versus females?
And this is the kind of be the guideline for what we're going to be discussing today. So it's going to get it might be a little confusing for you if you haven't been paying attention thus far, because you're going to have to use everything you know about chromosomes, alleles, how things are inherited to really understand all the answers to these questions. The state of Ohio expects that you're able to answer these on the end of course exam. And they're not going to give you a question like, what is gene linkage? What is in law of independent assortment? They're not going to do that. They're going to give you a problem, and then you're going to have to solve it understanding how this works. So you have to understand how this works in order for you to be able to solve the problem. That's what they do. And that's why I give you the kinds of questions I give you, because you need to be able to do that in a month or two. All right, so let's go ahead and take a look at what they're talking about here. I'm going to go ahead and just kind of, I think there's a way to just take this part, and then I can select it, copy it, delete it, move on to the next page. And I should be able to paste it. Yep, good. So keeping these discussion questions in mind, let's discuss some of the answers to them. So the first one is what additional complexities occur in humans and other animals that have to do with gene linkage, right? So when what happens to genes, and I answered this in the video as well when I discussed gene linkage and also dragon genetics. I repeatedly have said this throughout the whole process, and this is one of the reasons we went through meiosis to begin with. So you have this chromosome, and I really suck at drawing, by the way. It really bothers me that I suck so bad at drawing. These two sister chromatids, right? Let me start from the beginning. Here you have two of every chromosome, correct? Two copies. You're going to remember that every chromosome is actually a DNA molecule. Two strands, two polymers, two molecules of nucleic acids, deoxyribose nucleic acid bonded one after the other with a sequence. You're going to remember that there's all kinds of genes on here. And there's a lot of space in between that regulates those genes. So the pink is one gene, the blue is another, the green is a third, and they're all up and down the gene. So you see here that these three genes are very close to one another. This is very far apart. So these three genes are on a chromosome. They're going to move together. Once you remember during meiosis, remember what happens during meiosis, and you see it over and over again in dragon genetics, they get separated. They get put into a new nucleus, into a new cell. Correct? Yes? Chardon?
Yeah.
Yeah, okay. So these two chromosomes, you got one of these from mom, and you got one of these from dad. And I'm sorry that I'm using this pink and blue, because I know it's kind of passe to have pink being for girls and blue for boys. It's an old way of doing it, but I just think it helps emphasize and make it clear without question that this is that what we're talking about here is that you got this person got this chromosome from their mother, and the other chromosome they got from their father. All right, remember that you're always getting half of your DNA from the parental generation. That's a fact. And so when you're getting this gene, there's a gene. This allele could be big A, and the same allele on the homolog. Remember that these are homologs. I keep using the words because you're going to see them, meaning they have the same genes, but they might have different alleles. They could have the same if they're homologous, but they could be heterozygous, right? So in this case, they're heterozygous for the A, and then they have a B. Maybe they're homozygous for B, and for C, maybe they're heterozygous again for C, and then they have this gene. It's all the way down here. That's E, and they're heterozygous for that. So this is going to be true of all the chromosomes you have, all 23 pairs. So when you take a look at it, what happens during S phase of the cell cycle, right before they do meiosis, right before you made your eggs, or you gentlemen made your sperm? What is it that the cells had to do?
Replicates DNA.
Replicate the DNA is correct. So each of these have been replicated. In case you've forgotten, replicate means what?
Like copy.
Duplicate, copy, right? So that means that the DNA is the same. The versions of the genes are the same, correct? This is still big A. It's still in the same spot. It's a copy. When you photocopy something, they look identical if it works well. So they're identical. The sister chromatids are identical. Am I clear? All right. So the sister chromatids are identical at first. At first. Things get complicated. They always do. We don't make them that way. They just are. So, of course, the same thing happens with the father's chromosome because you copy all the DNA, not just mom's. And you'll get the same chromosomes happen, the same genes, the same versions of the genes, the same alleles on both chromosomes that came from dad as well. So that's what happens right before meiosis, and you split them up, and it's independent. They independently assort, except for these chromosomes because they're close together on the same chromosome. So these three are always going to move together. Why? Because they're on the same chromosome. If you separate, if this one goes into, remember what we're talking about, just to remind you that meiosis is you have this cell that has a homologous pair. They're copied, so there's 4 chromatids, 4 copies of the DNA. Then you have a split into 2 cells. The sister chromatids do not split at this point. That's not what happens. It's the homologs that split. And then they split again. Remember, there's 23 of these. I'm just drawing one for time's sake. Now you have 4 cells with 1 sister chromatid each. That's what's happening here. We're going to take these 4 and we're going to put 1 of each into these cells. Does that make sense? Does that make sense to you? You should all be taking notes, especially if you're in the room, there should be notes taken. Like I should see pencils and paper, things happening physically, right? So the independent assortment idea is that each one of these 4 is going to have a different combination. These chromatids will go wherever they go. But you'll notice because these are all on the same chromosome, these are always going to stay the same. They're always going to stick together. They're going to move together. Does that make sense? Because they're on the same chromosome. So you have this pink is going to be all the same. The blue gene is going to be all the same. Blue gene, get it? No, okay. The green gene is going to be all the same, and the yellow gene is going to be. So theoretically, they all move together because they're on the same chromosome. So if this is big A, it's always going to be big A on that chromosome, and that big A is always going to be around with big B, and that big A and B are always going to be around with C. And it's the same with the other combination. Little a and little c are always going to go together. Why? Because they're on the same chromosome. And they're all big B, so it's going to always be this combination together. This combination together. So that's what we call gene linkage. That is what gene linkage is, that the genes are close together on the same chromosome. So why not just are they on the same chromosome? Why not? Could you remind me of your name again, dear? I forgot. I'm sorry. Tamaya. Why not on this, just on the same chromosome? Why do they have to be close together on the same chromosome? What's the big deal with that? So we say that if genes are linked, if they're together on the same chromosome, why is that a big deal? Why do we worry about? Do you have any idea, Tamaya? Can you think of any reasons? You don't have to know. You can just make a good guess. So we talk about genes are linked. They're going to move together, but only if they're close. Why not? If they're on the same chromatid, they're on the same chromosome. Shouldn't they move together? What's going to stop them from moving together on the same chromosome? If they're on the same chromosome, what's going to stop them from moving together? Right? Like why isn't E also linked? Look at E. Why is E not also linked? Right, that's the reason we give you. But you're right on that. That's correct. Why is this one not always going to move with these three? So they're not linked. E is not linked, or at least not as linked, right? But because they're not close together is correct. That is the right answer. But what's going to allow E to move differently than A, B, and C? Why are A, B, and C always going to be together, but then E won't be with A, B, and C necessarily? Maybe big E goes with little a, B, and C, but big A, big B, big C are always going to be together. Why? Okay, so see, that's an interesting question, isn't it? It's not something you should know. Let me be clear on that. I don't expect you to know it. I'm just asking you to think about it. Does that make sense? All right, so the reason does anybody at home have any ideas? Anybody in cohort B or C have any ideas of why is it? And I explained this in more detail in the videos, but why are these three, why do we call these three linked? And this one all by itself, that's on the same chromosome. So if this chromatid goes here, shouldn't all four of these go here? Shouldn't this also be big E? How is it possible that these three big A, big B, big C could have a little e? How does that happen?
Could I guess?
Yeah, go ahead, guess.
So I guess since they're not like all linked together, they have the ability to use like the law of independent assortment. Like they can just move wherever they want. Oh,
Okay.
that be an
Well,
explanation?
that's not, remember what a law is. A law is what we see happen. So you're right that we do see that we get big A, big B, big C with little e, big A, big B, big C with big E. We get both versions. But why? How is that even possible since this chromosome has big A, big B, big C and the big E? How is it possible that you can get a little e here? Well, I'm going to tell you the answer. The answer is crossing over. Do you remember crossing over? All right, so crossing over is going to happen when you form this tetrad, especially in humans and non-dragons, right? Because the dragons was just an example organism. That's why we use dragons, because they're not real. So when we look at A, big B, big C, they're always going to move together. These genes are always going to move together because they're very close to one another. Crossing over has very little, it's unlikely that they're going to cross over in between A, B, and C because they're very close to one another. Does that make sense to you? I mean, don't just say, yeah, understand. The crossing over is that they randomly come across and they exchange DNA. If these two arms, these two sister chromatids randomly touch and switch DNA, which is more likely to switch in between A, B, and C and E, or in between A, B, and C and in between A, B, or C? If it's all random. between E and A, B, and C. Why? Because there's more distance between them. And it's more likely that you're going to get a split there. So we call that A, B, and C are linked, and E is far enough away that could break when it randomly happens. The break, you see how much randomness there is to life. If I try to find a color that we haven't used. So when this switch happens, remembering that this one you got from your mom, right? And the other one you got from dad. Also remembering that A, B, and C here, the A, the B, and the C here all go together as well. So little a, big B, and little c go together. They're linked, where little e is far enough away that there's going to be switching could happen between them pretty easily, and it's much more difficult for crossing over to happen between A, B, or C. So when we look at the break, and we see a break happen, so when this break, when this breaks here, those two break and they switch anywhere in between here, anywhere in this long span of DNA. Then they switch. Now you get a new combination. Now you get a sister chromatid that has the original, a sister chromatid that has a piece of dad's DNA, a sister chromatid is part dad's and part mom's, and a sister chromatid that's of the original as well. So you end up with, remember the centromere is holding them together, that you get, this one's still the same. All three of these are still the same because they're linked. The crossing over doesn't happen very easily in between them. You got this one's still B. It's still big B. And this was still C, little c, or big C, and this was still little c. These have changed somewhat. The C, E genes, this is still the original big E, but this was now little e because it switched. This used to be little e, now it's big E, and this is the original little e. So when we look at the chromosome, this is still mom's chromosome, the one that you inherited from your mother. This one, however, is mostly your mother's chromosome, but then you got a little bit of dad's chromosome got switched onto it, so the alleles are different. It's a different combination of alleles. This one's still mostly dad's chromosome, but then there's a little bit of mom's chromosome that got switched over. That's where that big E came from. And the other one's just a whole 100% dad's. So that when you see crossing that's why we say this is not, that E is not linked, because it doesn't have to move with A, B, and C. It's far enough away that crossing over allows it to move so that you could get big A, big B, big C with big E, which should be the original version. You could get big A, big B, big C with little e because of crossing over. You can get little a, little b, little c, little a, big B, little c with big E, or you can get it with little e. So some genes are linked and they have to be close together, and some genes aren't. So that's gene linkage in a nutshell. I go into a little more detail with the examples from the Dragon Genetics packet. And by the way, that's the answer to the last question before this section, which is, what are the two genetic inheritance big ideas that we talk about in Dragon Genetics 1? The first big idea is law of independent assortment. So if you don't understand, I don't blame you for not understanding. I need you to go back and look at the answers on the Dragon Genetics 1 and make sure you understand that, because that will be tested when you get back from spring break. Is that clear? And you'll have a take-home test as well. It's going to have you write an essay describing the law of independent assortment. That's part 1 of your take-home test. It's a simple, short thing in your own words. It shouldn't take you much time at all. But I need you to really think about it, and I give you the answer if you watch I just explained gene linkage. I've explained in the past law of independent assortment. In the video, I go into detail on it as well. So you have all the help you need, but you need to write it in your own words. Then the second part of that take-home test is to explain gene linkage. How does gene linkage work? Again, there's a whole video on gene linkage. I just uploaded. I'll post it as soon as I get you into your group work, and I just kind of outlined it here for you. Okay. So crossing over
Mr. Mendoza,
is something. Yeah.
Hussein said that his power got cut out.
That's okay. Tell them to join when we can, and we are recording. It will be recorded. Remembering that every lesson is recorded. All right. Tell him that, please, and thank you. So when we're looking at this, when we're looking at what happens that allows genes on the same chromosome not to be linked, then we're talking about crossing over. Now, do you think that now the next question, are all alleles completely dominant or completely recessive? Now that's going to take some thought as well. And let me see if I can just select this. Let me ungroup it. Let me do this. See if I can. And copy it and move another one.
Is it possible for like a linkage to like all of it move by crossover?
Good question. What a fantastic question, right? Yes. So the whole group of genes can move together. And in fact, when you watch that video on Dragon Genetics 1, that's the answer to that last section that's really complicated. You have to watch it because I really want to get into these alternate genetic methods before we go into spring break. And after today, we got a whole week. We're not going to see each other. You guys are going to relax, and I'm going to be relaxing. Actually, I'll be painting my wife demands that I paint the bedroom, so I'll be painting the bedroom. Spackling, all that stuff. So that's going to be part of my break as well. So in any case, I want to make sure I get this alternative inheritance pattern. So, but yes, the answer is absolutely yes. It's a great question, and I go into detail in that video that I'll give you in a minute. They're both an hour long. Both videos are an hour long. Let me be very clear. You don't have to sit down for an hour and watch a video. Does that make sense? If you understand it, if there's a button. I know this is a new technology, but it's there. It's called fast forward. Push the fast forward button. Skip it. If you miss something, go back. If you're really being efficient, look, you got a textbook that's this thick. Very few people read it from cover to cover. You reference a book, a textbook. Does that make sense? So you reference the video. Take what you need, leave the rest. All right. Are all the alleles completely dominant or completely recessive? The answer is obviously no. No, and let's talk about why no. Okay. We got to remember what genes are. Genes are what? What are genes? What are genes? Anybody? Just tell me what you can give me your idea. You don't have to look up any specific answer.
They're like instructions for creating people.
That's a very basic. That's a very generic, very ambiguous definition. So they are. You're right. They are instructions for creating people. They're instructions for creating trees as well. But that's you should understand this better by now.
Yeah, I just like gave like the most basic.
Yeah. They are not traits, but they're genes. They're sequences. They're instructions to help with the values definition, right? They're instructions you inherit from your parents. Is correct. So genes. But what else? What do genes do? Why? What's the rest of the DNA? Is all the DNA, the entire DNA molecule, is the whole chromosome a gene? No, only parts of it are genes. So what's the difference between the gene part of this DNA and the rest of it? What does the gene do that the rest of the DNA doesn't? You're going to kick yourself if I give you the answer. You should know that. This you should know. A gene is the portion of the DNA, specifically chromosome, that is transcribed. So it's the thing that makes RNA, right? That's all a gene is. The thing that you inherit, because just like Deidre said, that because it's transcribed, it leads to a phenotype. It leads to a trait. Just like Alexander said, that's how you get. That's how the instructions. Those are how you get instructed. The instructions are in the DNA, but how do you make the instructions work? Instructions mean you do something, right? What's the do part? If I tell you get up and go paint the wall, like my wife is telling me to go paint my room, now the instructions are go paint the room. The action is me painting the room. So the instructions of the gene are here in the DNA. What's the action? Transcription, which then for most genes leads to translation, which for most genes means a protein, some kind of protein. That protein could be a pigment like skin color. That protein could be eye color. That protein could be a hormone, a growth hormone that makes you grow. It could be another hormone that makes you, grow hair on your body. Another hormone that makes you grow more muscle on your body. And there's a lot of different proteins that do different functions. So if you have gene A and gene little a, if they're dominant recessive, most of the time you're going to see that dominant and recessive, they talk about is the gene dominant or is the gene recessive. It's really not. The genes are not really dominant or recessive, and that's an argument that the professor from MIT made, right? It's the phenotype is dominant, and the phenotype is recessive. So which is recessive, white or black? Which is recessive, red or yellow? Which is recessive, tall or short? It's not the gene. And the reason is, you'll see why, hopefully, by the time we're done talking about all the different options we have. So in dominant and recessive, you get a protein, gene A gives you, does transcription, which then does translation, and that protein leads to a trait that you see. Then gene little a does transcription, does translation. It wouldn't be a gene if it didn't do transcription. And finally, it leads to something. It's a trait. It produces a protein. Most of the time, there's a protein produced that's different. And you don't see it. So maybe there's enough of this protein that you're tall. Even though you have the gene for short, even though this protein would make you short because it doesn't work very well, you have enough of the tall protein, the protein that makes you tall, so that you're tall. In the case of peas, that's how it works. That's dominant recessive. Another good color, maybe another good way of looking at it is if you have a flower, maybe the trait that you produce makes you red, and the other trait leaves you doesn't produce a color at all. So the plant is, the flower is white. So maybe the flower is white. Well, if you have the gene for red, then you're going to be red. There is no pigment for white. The only reason the flower is white is because there's no color. It doesn't produce any pigment at all. In that case, then it's a red flower by default. Does that make sense? I keep saying that makes sense. People are nodding. I'm not so sure that's true, but I guess we'll have to see the quiz or the test. I'm not sure. So we call this dominant recessive. In other words, when you see this type of gene, this type of inheritance, if you get the version of the gene that gives you the dominant phenotype, in this case red, then you're going to get red. If you have big A, you got red. You don't see white at all until the next generation when you cross heterozygote. It's the same thing we talked about wrinkled peas. If you watch the MIT video, which was one of your assignments, your wrinkled peas versus smooth peas, we discussed it in class as well. So that's dominant versus recessive. The next type of gene inheritance that you can get. Now, what if you don't have? What if when you have A, if you go through transcription, you do transcription, and you do translation, but instead of the trait not showing up, what if you get two traits that show up? And you see it as well. Well, how could that happen? Well, what if you produce a pigment? Let's talk about pigment because that's the easiest to look. Instead of white being the result of the protein no pigment leading to a white flower, right? Versus a strong pigment dark pigment leads to a red flower, right? If you have the strong dark pigment, it's red. If you have the one that has no pigment, then you don't see it, okay? But what if you see it? What if you have a gene that you do have you produce a red pigment and you produce a white pigment? What happens then? What do you think might happen? What if you're producing a red pigment and a white pigment? What do you think the flower is going to be?
Could it be like spotted?
It could be, yeah. It could be spotted. Yeah, what's that? What was the other option someone said? It could be pink. That's right. The flower could end up being pink. So in the first, you had red. The flower's red because it was red pigment. The other gene didn't give you any pigment at all. So your flower was red. If you get two of these little a's in the first example, remembering that these are two different examples, the a and the a's are variables. If we used in the first example, the little a doesn't work. So you get big A, you get red. End of story. So that's how you get a red flower, because you have a little gene, a little a, and the little a doesn't really give you anything at all. And so the big A takes over. So in this case, there's two possible things that can happen if you're going to get both of them. You could have a flower that is pink. This is a mix in between, because you're mixing red with white. Now, instead of one gene not working, you're dealing with the possibility of the two genes the red and white mixing together to form pink. So that's one option. This is called blending. Now, in this case, the genes there's not dominant or recessive. They're not dominant or recessive. They're both kind of showing up and mixing together. So we don't have complete dominance. What do you think we call it if we don't have complete dominance? What do you think we call this kind of inheritance? It's not complete dominance. What do you think we would call it? When I ask you questions like this, it's because there's a chance you could figure it out. I'm not asking you to make myself look bright. What do you think we call something that does not have complete dominance?
Codominant?
Did you say codon what?
Codominant.
Oh, codominant. Yeah, that's a possibility. Let me go ahead and write that down. Codominant. And what's the another possibility? What else could you call something that's not completely dominant?
Like heterozygous?
Not completely dominant. Heterozygous. Yeah, it would be heterozygous. You're right. This is how you would get it. Is because if you're homozygous red, then you're red, right? If you're homozygous white, then you're white. But if you're heterozygous, then you're pink. But you're not red or white. You're pink. So you don't have complete dominance. What do you think we would call it? Partial dominance is a really good idea. I like that. Any other ideas?
Incomplete dominance.
And another idea is incomplete dominance. Any other ideas before we go on? All right, so incomplete dominance is the answer. Ding, ding, ding. Not that partial dominance is a bad one. I like partial dominance. That's a good way of thinking of it as well. But let's go ahead and get rid of that because that's not what they use. That's not what you're going to see on tests. So, but I think it's a very good answer. Incomplete dominance is when you have a mixture. That's when you mix the genes. The gene products, they both show up, but they mix together. And the details of the biochemistry are not important for you. There's another thing that could happen, that's codominance, and that's something Deidre said earlier. Deidre, do you remember what you said earlier is another possibility if they both show up?
Spots.
That's right, you can get spots. Codominance means you do get the red. The red does show up if you have it. And the white shows up. And the white shows up. So you get white flowers with red spots or red flowers with white spots, whatever the case may be. And the biochemistry is a little more interesting than this, but the genetics part of this, the idea is pretty straightforward. If you have the red gene, then the red shows up. If you have the white gene, the white shows up. Which one shows up where depends on, again, a little bit more complicated than what we're seeing here. So that's another way of inheriting genes. Sorry, I can't stand to make leaves that are white. Leaves have to be green. They don't work if they're not green. All right, so.
But I've seen plenty of bushes which are completely red or purple leaves.
Yeah, that's true. They do have red or purple leaves, but there's green in it. And you don't know, you can't see it, but the green is there as well. So there has to be green in if there's no green, it doesn't work for the most part. You can get red and blue and purple as well, but there's always green in it. All right. Does that make sense, guys? All right, so those are codominant. One's codominant, and of course the other one's incomplete dominant. So this incomplete dominance goes for when they're mixture, when there's a mix of colors. Codominant is when they both show up. And then you have here is complete, is either dominant, recessive. All right, let's talk about blood type because that's really a fantastic way of looking at blood type. By the way, when we're talking about dominant recessive, what you're going to see is this. It's either going to be A or big A or little a. We don't use those that nomenclature for codominant. All right, we're not dominant recessive. This is non-Mendelian genetics, so we're not using big A or little a. What we're going to use is big A, or let's use a different one, big R and W. Notice there's no little r, right? And up at the top, you have big A, little a. Big A is red, little a is white, right? Where incomplete dominance and codominance, you're going to use two different symbols because they're both showing up. So let's do Punnett square for the incomplete dominance really quickly. So if you have big A, big R, what do you got? What do you got if you have big R, big R?
Red.
You have red. So it's a red parent. What if you have big W, big W?
It's
You
gonna
have
be
a white parent. Let's see what their kids are going to look like. Here you have big R, big W. What color is this? You have red and white and incomplete dominance. What do you think is going to happen? Go ahead, make a guess. It's okay if you're wrong.
Would it
Anybody?
be pink? Because it's mixing white and red.
That's right. It's pink. That's right. Here, so this is R and W. So this is what? Pink. And this one's R and W. And so pink. And this is R and W. And this is pink. Oops. So you have a red and white flower coming together, mating, and they have 100% pink, I guess, pink progeny. Now with the codominant, we again have big R. So you have a red parent and a white parent. They come together, and what do you get?
White with red spots for all of
That's right. That's right. White with red spots.
Would it be red with white spot because the R is dominant?
Oh, they're both dominant. So they're codominant. So yeah, you can look at it. It's partially white, partially. In this case, the spots are red because I drew them that way. But in a lot of flowers, it'll just be there's a spot, there's a place here that's white, the place here that's red. The mixture could be all kinds of different shapes. So, but the bottom line is they're red and white. They're not pink. They're red and white, or white and red. So that's a really good question. Which is it, white with red spots or white or red with white spots? It depends on the flower. So that's great. And here's the cool thing about these. If you get a problem on the end of course exam that's codominance or that's incomplete dominance, it is so helpful. It is amazingly helpful. I mean, really, really, it's like, wow, this is easy. Well, why is that? Let's think about it. If you take an RW and you cross it with an RW, so you take two pink flowers, and the same is going to go over here with this case. There are no hidden genes. There's no carrier, no recessive. So when you get a red and white flower and red and white flower mixing together, you also get this result. Look at what happens. You get a red, 1/4 red, 1/2 red, 1/2 pink, 1/4 white. Easy peasy. There's no hidden, there's nothing about, oh, well, there's no question about what happened there. The phenotypic and genotypic, the genotypic ratio equals the phenotypic ratio. Do you remember us looking at the genes when we did a dihybrid cross with the dominant recessive, and we looked at 16 different options, and we had a 1 to 3 to 1 to 2 to 1 to 3 to 1 to 2, all that craziness with the genes, and it was a 9 to 3 to 3 to 1 ratio for the phenotypes, that they didn't equal. But this is real simple. This is really simple. The genetics here is really easy to see. You see, what you see is what you get. If you see a red flower, you know it's 100%, it's a homozygous flower, period, not dominant, because there's no dominance here. If you see a red-white flower phenotype, and that would be half, and this would be a quarter, or you see a white, and that would be 1/4, you know the white is homozygous W, and the red is homozygous for R. You know the genotype just by looking at the thing. That there's no math required. So it's really easy. It's really a wonderful experience to be dealing with codominance and incomplete dominance, because it's not, you don't have the same problems you have when you can hide it, when you can carry it. Everybody okay with that? We got that? Can I go on? I'm going to try to talk to you about red blood types. Blood types. An African American scientist in the 50s did a lot of work with blood types to help really develop it, and probably saved millions of lives during World War II and beyond. So we should, I forget the gentleman's name, but his work was foundational for developing this blood typing. Before blood types were discovered, it was virtually impossible to. Are you texting? What are you doing on your phone? Is it a game? I just need to know. It's one of those things, you know how inquiring minds need to know? Is it a game, or is it no, it's just texting? Okay. Because I don't have any, I see people playing games on there I watch, but I'm always like, how do you play a game on such a small screen? Why would you do that? But anyways, moving on. So blood types. Blood types do tell you something about the genes, but it's not as simple as dominant, recessive, incomplete dominant, or codominance. There's all kinds of things happening with blood types. So there's 3 genes, right? There's 3 alleles. There's actually 4 alleles. So you have this gene, one from mom and one from dad. You can have A, B, or you can have O. The A can be A, AB. It can be AO, which is A. And so A and B are codominant. A and B are codominant. So does that mean you get something in between? No. What does that mean? What does codominant mean? I just said it a minute ago. If you took notes, you should know. What is They both show up. That's right. They both show up. So let's take a look at the possibilities. What if I told you, and I'm going to go ahead and tell you. Now, I just told you that A, and I'm going to tell you this, I'll give you this clue as well. And you guys are going to work on it right now in class. O is recessive. So these are more than 2 alleles. A and B are codominant, O is recessive. Do we all understand each other? The phenotypes A and B are codominant, and the phenotype O is recessive. All right. You have O blood type. No, I'm sorry. Write it down, because I don't want to write it down. I'm not going to write it down. If you don't get it, then you don't get it. I think it's going to be fun, though. It's a puzzle. It's a Jerry Springer Maury Povich kind of problem. Are you ready? So you have A blood type A. Your husband has blood type B. You have your baby. You have a baby. He thinks you're cheating on him. And so he calls you out on Maury Povich. Is he still on Maury Povich? No. Who's that? What kind of show's out there like that? Give me something. What do you get? Nobody watches anything like that anymore? You know that whole drama, fake drama on the news, you know, you get a bunch of people on stage call each other cheaters, do DNA tests. Nobody does that anymore?
There are too many accidents that occurred after those things.
Oh yeah, people die, people get shot. I'll tell you what, there'd be some shooting if I were involved, guaranteed. You did what? And you put me on national TV? What? So let's go ahead and pretend, let's see, you're on the Deidre show, and Deidre gets you up on stage, and then you get sandbagged, and your husband's like, she cheated on me! So Deidre says, well, Jimmy, you married Jimmy, sorry. And Jimmy, well, Jimmy, we got your DNA test right here. You got your blood test right here. Your wife, whether you're male or female, you're now the wife. You are A blood type, and your husband is B blood type. But your child is O blood type. So go ahead, somebody figure out, did the woman cheat on the husband? Go ahead, work it out, and I'll give you 5 minutes and I'll tell you. You at home as well. The question is, did the woman cheat on the husband? Oh, you have an iPad too? Cool. You doing, Kira? What are you doing? Why aren't you in class? What you doing? Hello? Answer my question. You can hear about that. Step outside. Answer, you guys got 5 minutes. No, we're not stopping right there. You can stop right there. We're not gonna allow it. We're not gonna allow it. Okay, if you're doing something else, you move on. Take it, accept the fact that you move on. You clear? Don't lie to me. I'm not stupid. Okay? I don't think you're stupid, and I know I'm not. You and somebody else. Clear? Are you seriously gonna sit there and come here? I saw you. I was watching. I do not know if I'm watching. I literally was looking at your screen. You understand? This is so bad. you guys all this time? At home? Hello, Shark, are you there?
Yes, I'm
Rebellion?
here.
Did you all hear my question before I left?
No. Wait.
Oh,
Yeah.
are
Yeah,
you serious?
you are not the father.
Oh,
That's
so they
the
did
answer.
hear the question. Okay. Oh, okay. So you guys did hear the question. Oh, I'm sick of kids lying to me. I'm about to go karate somebody. All right. So what y'all think? How you defending yourself, woman?
You are not the
Did you cheat
father.
or not? Did the wife cheat or not?
Well, $50
I mean,
to
it's a
$50, so
Yeah,
use.
yeah. Okay, that's all right. It could have happened, but did the O blood type prove that she cheated? No, it didn't. Why? Why didn't it? Because it's recessive. That's right, because it's recessive. So let's take a look at that. By the way, people did. I remember a teacher who taught me biology told me a story. He taught his kids A and B blood type. They didn't understand what recessive meant, and their parents were A. Both their parents were A, and he was O. And so he convinced himself he was adopted. And his parents had to come in upset with the teacher. And the teacher's like, well, this is a result from not understanding what was taught because that's not what we said. So having different blood types is not what you're talking about with blood types is the phenotype. All right. So what we're talking about is you could have somebody with A, and here's what we do. We say IO. All right. So in fact, what we do is we say IA and little I for O. So this is someone who has a broken gene. By the way, O means there's no cell surface protein. Let's remember what a cell is. Let's remember that you have a cell that has a little, if it has this shape on it, it's A. And that's how your body knows it's you if you have A blood. Does anybody in here know if they have A blood type? You guys don't know your own blood type?
No. How
I think.
Your doctor should know, huh?
I asked my parents about it and they were like, we don't know.
It's good to know. You guys should know what your blood type is. I'm O blood type, by the way. Most people are O blood type. Thank God, and you'll see why in a minute. If it wasn't, I'd be in trouble. But about
Mr. Mendoza,
55-60% of the people are O blood type. Yeah.
isn't there another blood type, like it's called universal or something?
There's an RH there's something called an RH factor that's RH positive or RH negative. I, for instance, am O positive. That's just a smaller cell surface protein. So it's a protein that's on the surface of the cell. Your body sees it and says, okay, you're A, you're me, you're part of me. If they see a B, they're like, wait, yeah, that's not me, kill it. All right. So that's how that's what happened to people when they tried in World War II. People obviously got shot, they were hurt, they needed blood, they got blood. If they got the wrong type more than once, they ended up having an immune reaction and dying. Yeah, they could take it one time. Some people could take it one time. Some people couldn't take it all if it was the wrong blood type. But sometimes they would win. So doctors never gave blood to people in World War I and partly into II because they weren't sure what was going to happen. They had no way of telling what your blood type was. After that, on dog tags, you always have your blood type. So they always have your blood type so they know what blood to give you if you need it. So that's someone with A would have something that looks like that. I'm obviously making up the shape. I hope you realize that. And then B blood type, the shape is actually much more complicated. It's a certain shape of spaghetti. It's a long protein that twists and turns. I'm not doing that. So the other blood type B would look something like, if you want, I could pull up what it looks like for real, but Because today we can actually image them. We can actually see the molecule. On the cell surface of the protein, it looks something like this. I don't know why I do this. Why don't I just do something like that?
I have a question.
Yeah.
So, since A and B are codominant, could somebody like with A and B take A and B blood type?
You're a genius. You know that? That's an ingenious comment. You really need to consider that there are very few people actually think of that on their own. That's absolutely right. That's absolutely right. There are people that I tell that to, that get it wrong on a test, and you figured it out all by yourself. When you think differently, when you are able to come up with ideas, that's what we call genius. You realize that geniuses don't know everything, right? The geniuses are people that come up with innovative ways of looking at the problem. So yes, the answer to your question is absolutely right. Yes. So A has one shape and B has another. And obviously with AB, and that was not what I did, was not a genius move. It was not successful. So then with AB, what you have is both. So if you have AB blood type, which is going to speak to what you were saying, I was sort of exactly what I was going to say. If you have AB blood type, can I not draw the rest of this?
Yeah, it's fine.
Thank you. All right. So if you have A blood type and somebody gives you B, your body says, whoa, I don't got any pointy things on my cells. That needs to die. That's an invader, and it kills these things. If you're B blood type and you get an A blood put in you, you're like, hey, wait a minute, I've got any roundy things on my cells. You're dead. You're not me. But if you're AB blood type, it means you have both because they're codominant. If you're IB, you're AB blood type, then if you get A blood, your body's like, I got roundy things on my cells too. Hey, cool. If you get B blood type, you're like, I got little triangle things on my cells too. Okay, you're fine. So you can accept blood from A and B. If you're AB blood type, you're what we call the universal. You're the universal acceptor. If you're AB blood type, you can take blood from anybody. That's another genius question, right? Another. Oops. Wait a minute. O blood type. We got, and I say we because I'm one of those people, and that's most of us. We have genes that don't produce any cell surface protein. If you're O negative, you don't even produce the little Rh factor. You have surface proteins, but nothing that makes your body react. So when you're used to seeing nothing on the outside of your cell. So if you have A blood given to you, you're like, I don't have any triangle roundy things on my cells. You're dead. My body says, oh wait, I don't have any triangly things on my cell surfaces. You're dead. If even both AB blood, if I get AB blood, my body's like, wait, you got triangles, and no, you're dead. You're not part of me. I have nothing on myself. So, but interestingly, what else can you think about that? What would we call? We're a universal what? What can we do? We can't take anybody's blood, but what can we do? We can give. We can give our blood to anybody. Why? Doesn't have any. There's nothing to react to. So my blood I can give to A, to B, to AB. My blood is prized by the vampires that we call the Red Cross. They want my blood very badly because I have O blood type. O negative is the most sought after because they can give to anybody because I can only give to people that are O positive. They are positive. They have a tiny little protein, tiny little protein on the cell surface called RH. It's called an RH factor where some people have it, they're AB positive, they can be AB negative, they don't have the RH, either have it or you don't. So there's no dominant, codominance going on there. It's just either you have it, it's a dominant or recessive. So when you're looking at the different possible combinations, you can either you inherit two genes, the I gene, it's either IA, you inherit IB, or you inherit little I. Little I, we call it O blood type. And in the case of our mystery guest on the Deidre show, that person would have had, the parents would have had little a, IA, little I, and they gave to their child what? What did they give to their child? They're going to give genes. The two little eyes. Yeah, that's right. So they have one of the parents had a little I. Another one of the parents had a little I. And so some of their children are going to be A blood type. Some of their children are and only 1/4, but it's still possible. Some of their children will have O blood type. Now, what if that child would have had B blood type? Then there was some cheating going on. So you can use blood type to help to rule out certain outcomes. You can't really prove anything with blood type because of the recessive little I. But you can if the blood types are, if the blood type is something you can't make, like you can't, if you're A, whether you're AA or AI, little I, no matter what, you can't make a B unless you marry somebody that has B blood type. Now, if you married someone who's AB, then that would be possible, right? So somebody who has IA, IB, and you marry somebody who has A blood type, could you have someone with B blood type? How about that? What if the parents were AB blood type and A blood type? Could they have made a baby with B blood type? Was there cheating going on? Yeah, they could. How? And there is some codominance. So AB is codominant, so they would look like this. And the other parent has A blood type. How can you make a B? The baby has to have B. How is that possible? That does. This parent has a B. The other parent has A. How do you get a child with B? With just B? That it's because of that recessive. It is that not incomplete, but because the B is dominant over the recessive little I. See how confusing it is? Yeah. So these two people could have a child with A blood type, a child with AB blood type, or a child with a B blood type. The one thing they could not have is a child with O blood type. There's no way, because one of the parents doesn't have the little I. So remember, you can only get what your parents give you. If your parents don't have it, you can't get it. That's how genes work. That's how inheritance works. Okay. It's 10. It's almost 10. I'm going to go ahead and start playing the video on this screen and let you take notes on it. Is that a good idea? Are we going to start that? Oh, you know what? Go ahead and give you the take-home test. So the take-home test that you have to do by end of break. You have 2 essays to write. The first essay, explain the law of independent assortment. That's it. That's all you gotta do. And using, now I need to see an example. And it has to be in your words. You can work together as a group. I want to warn you right now, I will be checking your answers for plagiarism. We have an app, an app that searches the internet for anything you might have cut and pasted. And it tells us how similar it is to anybody else's work. So do not copy and paste, because I will know. The machine will spit out, hey, this work is, all you gotta do is copy your work, right? You're gonna turn it into Schoology. The question will be put in Schoology as a test. You're gonna put in your answer. I'm gonna copy it. I'm gonna paste it into the online software base. It'll search. If it matches 99, 98%, right? Then you probably copied and pasted, right? So don't copy and paste. It can tell the difference between your writing and someone else's writing, even if you use fancy words. It's not the word, it's the usage of the word. It's how you phrase things. Everybody phrases things differently. But you can be similar to each other. You're allowed to be similar to each other. You just can't be similar to, as long as that you tell me you're working as in a group. Is that clear? So you can't, I encourage you to work in a group. So explain the law of independent assortment. And the second one, explain gene linkage. Again, give an example, just one example, and in your own words. This does not, the length, I always get questions about length when it comes to essays. It can be as long or as short as you want it to be. I'm not putting any length requirements. I just need you to make sure that the answer is complete. Do you all hear me? Answer completely. I don't care how long it takes. It could work. Look, you could. I don't want you to turn in 2 pages worth of discussion because I don't want to read it. But if you do two pages, it's well done. Great. You could do. I could literally do this probably in a paragraph. I'll be honest. The shorter it is, the harder it is to do it well and do it completely. It's easier to write more and be more complete. It just takes a lot more effort to write less. I don't care what you do. Whatever works for you and your friends. Get together, answer these 2 questions. You're done for this class for spring break, and get it done before Friday if you want. Okay? I don't want to put any stress on you. I just want you to think about these 2 ideas of gene linkage and independent assortment. The videos that I'm about to post on Schoology, have those, discuss it very well. I know because I'm the one who's discussing it.